Hi, my name is Hiroki R.Ueda. Please call me Hiro. Today I'm going to talk about the whole brain and whole organ imaging with single-cell resolution by tissue clearing method called QBEC. So, as you may know, tissue clearing as well as light sheet microscopy allow us to observe every single cell existing in our brain. So let me introduce SODA, state-of-the-art in this field. For example, in 2018, we succeeded to observe the whole brain profiling of cell existing in the mice adult brains. And also, just recently, we succeeded to observe every single cell existing in the newborn mice, which is about 428 million cells. So if you're interested in this field, please starting with our recent review published in Nature Review Neuroscience as well as Newton. Physics of tissue clearing is already proposed by Dr. Spaltholt in 1911. So first, we try to minimize light scattering by homogenizing refractive indices of each substances. Second, we try to minimize light absorption by removing the pigments such as heme from cells. So there are already a couple of pioneering works. For example, hydrophobic reagent have been introduced by Harald Lundvall in Sweden and also Werner Spaltholt in Germany. And after more than 80 years later, hydrophilic reagent was introduced by Professor Tooting and his colleagues. So far, there are three main categories in tissue clearing method: hydrophobic method, and also hydrophilic method, and hydrogel-based method. So there are pros and cons among these three categories. We focus on that second hydrophilic based method, reagent-based method, because it has a high scalability and a higher safety, but relatively low performance. So therefore, we try to improve the performance in hydrophilic method. So we took a chemical screening strategy to identify the potent hydrophilic chemicals. So, but if you want to use one brain or one organ for one chemical, it takes a lot of time and very labor intensive, and also we have to sacrifice a lot of animal lives. So therefore, we use a brain paste, and then we use a small portion of the brain paste, and then perform the chemical screening, which works pretty well. In 2014, we succeeded to identify the series of chemical substances, including amino alcohol, which have a very potent property to removing the lipid. By using these reagent together with the detergent, we succeeded to make adult mice brain clears. And we also identified that same chemical are potent to eluting the heme from red blood cells. So therefore, we use these methods not only brain but also entire mice bodies, and which works pretty well. So at that time, we speculated that interaction between heme and hemoglobin, which is mediated by histidine and heme interaction, seems to be competed with the nitrogen existing in the amino alcohols. So after we clear the samples, we can use light sheet microscopy, which have a unique setup. For example, we illuminate the sample from the side and then observe the sample from the top. And then if you change the height, we can take the picture one by one, and then reconstitute the three-dimensional structure of the samples. So by using this method, we can conduct three-dimensional anatomy. For example, heart, lung, kidney, and also liver can be visualized with the single-cell resolutions. We can also apply same method not only to anatomy but also to pharmacologies. In this case, we can compare the control mice and also NMDA receptor inhibitor administered mice, which seems to have schizophrenic phenotype in mice. And as you may see here, you can see the hyperactivation of the cortical activity of the neurons, which is visualized by the immediate early gene called ARC. Okay, so physics is already available, but how about the chemistry? Unfortunately, at that time, around 2014, chemistry of tissue clearing is totally unknown. For example, we show the decolorization of heme is possible in very mild condition, but nobody knows the chemical foundation of it. And delipidation is possible with the amino alcohol, but we still don't know the potent key functional group of delipidations. Decalcification is partially possible, but maybe we can improve it. And also, underlying chemical principle is unknown. And then refractive indices matching is interesting, but we still want to know the functional group for it. And then sometimes expansion of the tissue can contribute to that transparency, but we still don't know the functional chemical group for expansion of the tissues. So to analyze the chemical properties and also chemical structures, we performed the chemical profiling of more than 1.6 thousand chemicals. And then we conducted 5 different assays, including delipidation, decolorization, decalcification, and also expansion and RI matching. And then we found a couple of potent chemical as well as a functional group, which is important for each property for the elementary tissue clearing process. So if you use or combine delipidation and also RI matching, we can perform the whole kidney profiling of every single glomeruli existing in the kidney. So in this case, we identified that 12,000 glomeruli existing in the mice kidney. And also, we can apply same technique to the pathology, like a cancer metastasis, which is a kind of a collaboration with the cancer biologist, Professor Miyazono. And then we try to look at the cancer metastasis spreading all over the mice bodies, visualized in the emetry signal in here. Okay. So as you can see here, you can see the spreading of the cancer cells. We can also apply same tissue clearing technique not only to rodent but also to human, especially cancer metastasis. So we collaborated with Dr. Nojima and also Professor Mali to observe the three-dimensional structures of the human lymph node with the cancer metastasis. Okay, so by using tissue clearing method and also three-dimensional observation, we can increase the sensitivity to detect the cancer metastasized to human lymph node. We can also apply tissue clearing and also three-dimensional profiling to primate. So in this case, we try to look at the marmoset, entire marmoset. And also, by using the potent chemical, we can also apply this tissue clearing technique not only marmoset, rodent, but also to the variety of the tissues, including heart, liver, spleen, kidney, brain, lung of human. Okay. So after we publish these papers, there are a lot of publications, not only us, but also other people's. One of the unexpected applications come out recently, which is the application to the pill bag to make a transparent pill bag, and also to make a transparent crab. And then, much surprisingly, Japanese high school student Monto Kuroda tried to use our technique and slightly modified and apply to drone beetles and horn beetles and stuck beetle to make transparent beetles. And he got the prize by this work. Okay, how about the bone? So bone clearing was a little bit problematic. So EDTA was already used for many years, but potency of bone clearing is limited. So we screened the chemical, and then we found out that not only calcium chelation, but also protonation of the counter ion phosphate seems to be important. And also, this can be mediated by the imidazoles. And then by using this technique, we can visualize bone metastasis. Okay. Now, how about the decolorization, especially decolorization of the heme in red blood cells? So we screen the chemical, we find out 1-methyl-imidazole seems to be one of the most potent chemical. Interestingly, this looks like a histogen, which mediate the interaction between the heme and hemoglobins. So our hypothesis that nitrogen in amino alcohol might compete with the interaction between heme and histidine is now supported by the second chemical profiling and then finding of the 1-methyl-imidazole as potent chemicals. How about the expansions? So our grad student Tomoyuki and also Tatsuya tried to look at the every single cell existing in the mice brain, but the resolution is limited. So therefore, they try to screen the chemical which can expand the fixed brain. So we identify the imidazole and antipylin seems to be very potent to expand the brain. So therefore, and then Tomo constructed optimized light shade microscopy equipped with the lens of 0.6 Na 10x and working distance 80 millimeters. So he tried to look at the entire mice brain by repeating the observation. So we use 400 stacks to reconstitute the entire mice brain, and then look at every single cell existing in the mice brain, which end up with 100 million cells. And then actually this is a kind of a larger population than entire German populations. During this procedure, we also notice a very interesting physicochemical property of our brains. Actually, our brain acts exactly as polymer gel or hydrogel, which is consistent with Professor Tanaka's idea in MIT. So to validate this idea, we change the pH and also osmolarity and also dehydrations. And then we found our brain expand, reversibly expand and shrink, and reversibly expand and shrink again, which is actually property of the hydrogels. So if our brain or tissues are hydrogel, we can use a very old equation called Lorentz equations to guess the property of the refractive indices. And also, so by using this concept, we can summarize our finding. For example, delipidation decalcification effect on the refractive indices of the monomers or unit, and then expansion effect on the volume of the unit. And the composite refractive indices should be matched with the medium by using RI matching of the mediums. And then minimize light scattering and also minimization of the light absorption, which is achieved by decolorization, together achieve the higher transparency. And then interestingly, we also find out the important chemical functional group, which is essential for the RI matching, delipidation, decalcification, expansion, and decolorizations. So if you're interested in, we describe these logic in our recent reviews. Okay. So now we have a profound principles, and then we feel it's high time to move forward to the biological application. So therefore, we constructed the single cell resolution brain atlas. And then by using this atlas, we can map cell type or cell activity or cell-cell connection onto the atlas to look at to investigate the biologies. For that purpose, we also constructed the three-dimensional antibody staining method called cubic HP, and then now we can apply a lot of antibody staining to the entire brains. Okay, and also these techniques are available to the commercial companies. And also we constructed the cloud-based software allow the user to use or visualize the entire brain atlas as well as use their own data. Okay. So for example, by using entire brain immunohistochemistry as well as cloud-based software, we can look at the PV neurons, somatostatin neurons, or cholinergic neuron, or catecholaminergic neurons, and also brain activity, as well as brain cell-cell connection by using the rabies virus. And then Cubic Cloud software is available recently. Okay, so what is the futures? So there are at least three directions. So first direction to be faster. So we now try to reduce time which is required for the observations. So 3 hours for 1x microscopy should be reduced less than 5 minutes. 3 days for the 10x microscopy should be reduced to 4 hours. And the next direction would be smaller. So we now try to look at the entire synapses in the mice brains. So we call this project as Petascope project because 1 petabyte data can be given rise in this produced in this project. And the third direction would be bigger. So we try to achieve the kind of a large sample observation by making the appropriate microscopies. And then now we can succeed to clear the entire marmoset infant bodies and also adult marmoset brains. And also we recently succeeded to stain the hemisphere of the adult marmoset brains. So combining these 3 directions, we now trying to make an organ atlas. So we recently introduced axially sweeping technique into our high-speed light sheet microscopy called MOVIES, and then we constructed actual movie microscopies. So by using this, we can now making an atlas of kidney and heart and lung, and also as I introduced, a whole body of newborn mice. Okay, so today I present our work, but this effort is totally dependent on the collaboration with our colleagues and with our collaborators. I'd like to thank our collaborators and the colleagues, and I'd like to thank you for taking your precious time to watch my talk. And if you are interested in becoming a first transparent human, please email me. We call for the human samples. Okay, so I'd like to take any question from you.